Alright, ladies and gentlemen, this is going to be our next video on photosynthesis, specifically looking at leaf structure. So let's get started on this next video. So let's say I take a microscope and I zoom in on this leaf. If I zoom in on this leaf, maybe this is 40x, it's one of the lower powers. What I'm going to see is this intricate pattern on the cuticle of the leaf. Then if I were to zoom in a little bit further and take a cross section of the epidermis of this leaf, what I'm going to see is all these different types of cells that exist here. So the goal here is to kind of delve into what those types of cells are and what function and role they have in the leaves. Now, the first thing we have to think about is what is the purpose of leaves on plants? Well, we know that leaves are going to go through and collect sunlight to go through photosynthesis, but leaves are designed to have as much possible surface area in order to enable for maximum collection of solar radiation. So they're designed and they have evolved in a way that allows them to spread themselves out and capture as much light intensity and sunlight as possible to make sure that they're going through and producing that glucose. Remember, we're taking that carbon dioxide and that water and we're using the energy of the sun to convert it to glucose. So we're going from light energy into that chemical energy which we store in that glucose. So it's really important that we have a lot of surface area to capture as much sunlight as possible. So if we look at this cross section of a leaf, this is the top of the leaf here. So it's going to be the top. And then this would be where the bottom of the leaf is here. And what we've done is we've taken a cross section and we've kind of taken out chunks here to go through and look at all the different types of cells that are in this particular leaf or commonly in leaves that we see. So the first thing that we're going to have to go through and look at is what we call the cuticle. So this top part we call the cuticle. And this is really going through and it's creating a barrier to not only not let water in, it's hydrophobic, but not let water out. It's regulating homeostasis and ensuring that the leaves go through and maintain water for all the photosynthetic cells here, and then maintaining water through the xylem and phloem. Now, if we go and break it down into tissue layers, we can break it down into the upper epidermis here. See the upper epidermis. And then we also have our lower epidermis here. Epidermis. And these are tissue layers. So we call this the upper part of the leaf and then the lower epidermis of the leaf. And in the upper dermis, we have what we call palisade cells. And these palisade cells are really, really important for photosynthesis. These here are the cells that contain a large amount of chloroplast because remember, the light is coming down and it's creating electromagnetic radiation, the sun is, that's going through and powering photosynthesis in the electron transport chain. So all around here, there's lots and lots of chloroplast because this is where the top of the leaf is. If we go down to the bottom, we have what we call our spongy mesophyll cells. So spongy meso, P-H-Y-L-L. And you can see that these palisade cells are really, really tightly compact and these are not. And the reason that is because we have these guard cells or these stomal cells that go through and let carbon dioxide in. Now, as carbon dioxide travels through here and gets in through this opening or this pore, it needs to go through and get to the palisade cells, which are here. And it has to go through and it has to have a root. So in order to get to all the palisade cells, the spongy mesophyll cells are really, really spread apart because it allows for the carbon dioxide to go through and travel in between the cracks of that spongy mesophyll. Now over here, what we're going to see is our xylem and our phloem. And these go through and transport sugars and water. This is what we call our vascular bundle. So our vascular bundle, and they enable water and transpiration to occur in the actual leaf. So this is kind of like the veins in your body here, and those veins enable blood to move through. Well, plants need to have water moving through them, so this goes through and enables that water to move through the actual leaf. So the first type of cell that we're going to look at are these palisade mesophyll cells, and these are really going to be the cells at the very top section of the leaf here. So the entire function of these cells is to contain the chloroplasts. And the reason they're at the top of this leaf is in order to go through and capture the sunlight. All the sun's reaching and hitting the top of the leaf, so it's really, really important that these chloroplasts are located at the top of the leaf in order to maximize solar retention. Now, if we delve in a little bit more, what we're going to see at the bottom are these stomata, or these guard cells. When we talk about guard cells, we're talking about one half of the cells. And they let carbon dioxide into the cell. Remember, carbon dioxide is one of the reactants that we need to make our glucose. Now, they're going to look like this. They kind of look like two lima beans. And dependent upon turgor pressure, they can open or they can close. And this is really controlled by these sodium-potassium pumps that we've gone through and talked about quite a lot already. Now, the entire purpose of these stomatal cells, which we can see if we were looking at a real cross-section of leaf or here, is to allow for transpiration, the pull of water into the actual leaves. So they're creating a transpiration system that's allowing water to move up through the roots. And they're also going through and allowing carbon dioxide, the reactant, to get into the actual cell of the leaves, into those palisade cells. So the other thing that we have to think about is plants are also going to need the water. So we can limit and control water loss by opening and closing these cells. So when they're closed, the stomata cells can go through and prevent water loss. So that way they can conserve their water for photosynthesis. And then when they're open, they can go through and allow for more water pull up through the plant. And they can also go through and enable for an increase in carbon dioxide. Now, the next type of cell is going to be these spongy mesophyll cells. And these are cells in the large spaces between all of these cells. So we're going to see them at the bottom. And the carbon dioxide is going to travel in between each of these spongy mesophyll cells to reach the palisade cells. That's the reason that they're so spread out, is it creates space for the carbon dioxide to move as they come in through the stomata. Now, if we were to look at them, they're going to be located on the bottom half of this leaf. So the top is where we have the cuticle, palisade cells. Then at the bottom here, we have our spongy mesophyll cells. We can see that they're much more spread out. They're not as dense because the carbon dioxide has to travel through all of those spaces. Now, if we look at this a little bit more, what we can see is water is going to move through a vascular system, which we call our vascular bundle. So if we were to look at the roots of a plant, well, we're going to use transpiration to pull the water up through the roots. But the vascular bundles, the xylem and phloem, are going to allow the water to travel through this system of plants. Without this vascular system, well, the water wouldn't be able to get to all the necessary cells to go through. And remember, it's a reactant for photosynthesis, but get to the palisade cells and allow for photosynthesis to occur. So the vascular bundle is just like the veins in our body. It's allowing for the transpiration of water, the movement of water through the roots in joint contact with the stomatal cells, and allowing that water to move throughout the actual plant. Now, lastly, we have the waxy cuticle, and this is a water impervious protective layer that goes through and covers the epidermal cells. So what it does is allows and regulates water loss. Water can't really get in. It can only get in through the stomata or through the roots, and it's going to prevent water loss, so water getting out of the cells so the leaves don't dry up, so that way they can regulate homeostasis and regulate photosynthesis. Remember, this is just another way that the plant is regulating homeostasis. So the waxy cuticle is really, really important for maintaining and then providing support for all of the different water systems that move in through the plant. Now, there are going to be a variety of factors that are going to affect photosynthesis. There's going to be light. There's also going to be temperature. We're also going to have carbon dioxide and then water. Remember, carbon dioxide and water are the reactants that go in to form the glucose. We need the carbon, the hydrogens, and the oxygens to be rearranged to form that glucose. And then light is providing the energy, and then temperature is going to affect the enzymes which complete this chemical reaction. So if we're looking at rate of photosynthesis, well, let's think about light intensity. The light is going to be going through and emitting solar radiation on the chloroplasts, and the higher intensity light excites electrons more in the chlorophyll. When it excites these electrons more, it's going to have an effect on the rate of photosynthesis. So what we're looking at is light intensity versus rate, and the more light that I get, the higher the rate of photosynthesis. Now there comes a point where this tapers off, and that's where we kind of see this plateau on that graph. Eventually, there's only going to be so much light that the photosystems can take in, and then because of that, they can only maximize so much of the energy going into photosystem 2 and photosystem 1. So because of that, we actually see a taper at the very, very top end of this graph because there's a certain area, or there's a certain amount of light that will eventually no longer increase the rate of photosynthesis. Now, another factor that's going to affect it is temperature. We've talked about temperature a lot in this class. Temperature is going to have an effect on the enzymes. So the chemical reactions that occur during this chemical reaction are controlled by enzymes. Higher temperatures are going to accelerate chemical reactions, but too hot of temperatures are going to cause the actual enzymes to denature. So what we have is this Goldilocks curve here, and we can see that if the temperature does get too high, the enzymes will denature. Remember, they're going to go through and they will fall apart. But also, if it gets too cold, the enzymes can't function. So there's a happy medium, generally closer to the warmer side of that chemical reaction, that allows for the temperature and the rate of photosynthesis to be in harmony and allow for photosynthesis to occur at a maximum rate. But again, not too hot. We don't want the enzymes to denature during photosynthesis. Now, carbon dioxide and water are the two reactants that go into this chemical process, and they increase the rate of photosynthesis to a point, carbon dioxide does, and then it levels off, just like light intensity. So the more carbon dioxide there is, or the more carbon dioxide available, well, it's also going to go through and increase the rate of photosynthesis. But again, there's only so many enzymes that exist in this complex or in this chemical reaction. So eventually, they're not going to be able to go through and handle all the photosynthesis coming in. So that's why we see that tapering. Same thing with water. So water is going to go through and split the electrons from the actual water molecules, and we're using that energy down the electron transport chain. So this is going to increase the rate of photosynthesis as well to a point, and then it's also going to level off. All right, so did you guys learn? Did you learn a couple of things? Did you learn the leaf structure and parts of the actual leaf? Did you kind of get an introduction to light and dark reactions? I've kind of brought up the electron transport chain a little bit. And then did you guys go through and review photosynthesis? This is going to be the end of the video. We'll see you guys in class tomorrow.